In the not-too-distant future, a fungus called cordyceps will mutate in response to a warming planet and evolve the ability to infect humans. The fungus takes over its victims' brains, turning them into zombies hell-bent on consuming every living person they can find. The resulting panic will create a dystopian world where our survival is far from guaranteed. Sounds bad. Lucky for you, that's just the plot of The Last of Us and not a prediction of the actual future. As far as we know, anyway. But if you played the game or watched the show and have been worried about cordyceps becoming a reality, I do have some bad news for you, also some good news, and then some really bad news. The bad news is that the cordyceps fungus is real and does cause zombie epidemics. The good news is that it mainly targets insects and spiders, not humans, so you don't have to worry about it for now. But the really bad news is that there are plenty of other fungi out there that have the potential to become a disaster for us. And fungal infections are on the rise, which is why researchers like Asiya Gusa are so important. Her work to understand how fungi adapt to changing environments and what they could mean for our health can help us prepare for whatever they throw at us. We sent our friend, Madelyn Leembruggen, to Duke University to tour Gusa's lab and get a first-hand look at the potential threat. Get ready for another SciShow field trip.
Thanks, Hank. He's such a fun guy. Stop me if you've heard this one before. One day, totally out of the blue, a new pathogen evolves from something more benign, and this new bug is very good at making us sick. It's highly contagious, we don't have great medicines or vaccines for it, and the sudden surge of sick people throws the entire world into chaos. If you're having flashbacks to sanitizing our groceries in 2020, well, I don't blame you. But when we talk about pandemics, we tend to think of viruses or bacteria, things like the flu or COVID or tuberculosis. What we usually don't think of are fungi. Fortunately, Asiya Gusa does. Gusa is an assistant professor of molecular genetics and microbiology at Duke University, where she spends her days studying fungi that have the potential to cause dangerous infections. So as you can imagine, she talks a lot about fungus. I noticed that you said fungi. I say fungi.
Fungi, 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 however you want to say it is actually okay.
Okay. Is there like debate in the community about, or everyone just kind of picks their
We'll talk
favorite?
about whether or not you want to use a hard G or not. But yeah, I've landed on fungi, but whatever makes you comfortable.
One of your body's great defenses against fungi is the fact that you are, well, kind of hot. And I mean that literally. So why are fungal infections not very common in humans?
It's a really good question. And we think that it's actually, a lot of it has to do with our higher body temperature. So our higher body temperature serves as a natural kind of thermal barrier. Since most environmental fungi that we breathe in the environment, they actually can't grow that well at our body temperature. They prefer cooler temperatures, like around 25 to 30 degrees Celsius, because they live in the soils and they live on plant matter. So we actually have sort of a natural defense mechanism of our higher body temperature.
But there's a group of fungi that have the potential to change that, which is why Gusa and her team are studying them. They're called cryptococcus. These fungi are already known to cause serious infections in some immunocompromised people. That's because even though our body heat holds most fungi at bay, our immune systems still have to finish them off. And as the planet warms, cryptococcus might be adapting to survive better and for longer at the higher temperatures that usually keep them out of all our bodies.
So actually, in the laboratory, we study a species of cryptococcus that's less thermal tolerant. So it can't survive well at body temperature. And so those are the ones we're most interested in studying and figuring out if they cross that temperature threshold, they are possibly going to cause a greater number of disease, increase in incidence, and also severity of disease.
Organisms are constantly adapting to the world around them, and some of them, like cryptococcus, are very good at doing that. Like all living things, these fungi are always mutating, sometimes based on random changes to single base pairs in their DNA. These little changes can add up over time, creating big alterations in how the organism functions.
What we're in right now is a big 30-degree incubator, so think of like a walk-in fridge, like a cold room, for example. Same thing, but the opposite.
That's Cheyenne Lee, a postdoc working in Gusa's lab.
The reason that we have this here basically is the cryptococcus cells that we grow primarily grow at 30 degrees Celsius.
So in this room, they're not feeling the stress.
Yeah, they're not feeling that heat temperature stress. So that's the idea. So these are just some little crypto cells on a plate, and then I have another one that was previously grown at 37 degrees. So you'll notice that there's a lot fewer colonies on the plate because they were very stressed out at that higher temperature.
I also get stressed when it's too hot.
Me too.
In addition to getting stressed in the heat, something interesting changes about the way cryptococcus mutates. Gusa found that the mutation rate is connected to the temperature around the fungus while it's growing. When cryptococcus was grown at 37 degrees Celsius, it mutated 5 times faster compared to when it was grown at its more typical 30 degrees Celsius. But what Gusa found even more interesting about her studies was how cryptococcus was mutating. These weren't just single base pair changes that were accumulating. There were these big chunks of DNA jumping around, called jumping genes, or transposable elements. These transposable elements can have a big impact on how genes are expressed. And I even got a hands-on look at how they studied these jumping genes.
What I'm going to actually show you guys today is how do we check to see, well, has that jumping gene actually jumped? So that's what I'm going to be looking at here.
We're going to see how the jumping genes jump. We're about to do a test called gel electrophoresis. The researchers isolate a segment of DNA, chop it up, and stick it on a gel plate, which they run an electric current through. This causes the DNA to sort itself by size, and those bigger pieces are where the jumping genes landed.
So if you want, do you remember how I loaded that at the very beginning? You've been watching a couple of times?
Yeah.
Do you feel brave?
I can be brave. I can be brave.
All right, so what I'm going to have you do is load one of those. There you go. Wonderful.
That back to you.
Yeah.
Okay, and then right next to where you put yours?
Or wherever you want, really. And then, that's perfect. Beautiful. Oh, get it towards the end and then put it. There you go. Now press the rest of it in there. There you go. Perfect. All right, that should be enough. You got it. So now the next button that you can press is right there. Press that. Yay! Yeah! Great job! Yes!
Combined with the rapid rate of mutation, these jumping genes can lead to the kind of heat tolerance that some species of cryptococcus have shown lately. As if that's not bad enough, Gusa found that this higher mutation rate can give cryptococcus a really nasty superpower: drug resistance.
So these are the drug-resistant colonies, basically on both of these plates. 37 degrees, these were the ones that were stressed out. You can see, just like on the other plates, there's a lot less.
And what we're looking at are spontaneous drug-resistant mutants.
Yeah.
So if you grow up a population of cells, a small percentage of them will have a mutation that will allow them to grow on a drug-containing plate. And so this is kind of a proxy for us to figure out if there's been a mutation that has caused a particular phenotype to arise, which is drug resistance in this case. So then we can actually go in, extract the DNA, and find out what that genetic change has occurred. And in some cases, we find transposable elements have inserted into our gene, causing the drug resistance.
Even the drugs that currently exist for cryptococcus aren't great. The ideal treatment involves multiple antifungal drugs, which are often toxic and difficult for people to handle. Gusa told us that one of the biggest challenges in treating these more serious fungal infections is that there just hasn't been a lot of research done to improve these antifungal drugs.
And so what we really need are better drugs to treat fungal diseases and just to be prepared in case we need fungal vaccines. Right now, there are no fungal vaccines to treat humans. And so that's a big concern. And then a lot of the drugs that we have to treat fungal infections were very antiquated. They were developed a long time ago. They actually have a lot of toxic side effects. So more investment is needed in developing effective drugs and drugs that people can take, potentially orally to treat fungal infections that are invasive.
That's another potential benefit of Gusa's research. If she can identify the conditions and substances that kill certain fungi, it could lead to promising antifungal treatments. For example, the fungus that makes penicillin, Penicillium, makes its own antibiotics to reduce resource competition in its environment by killing nearby bacteria. Why is it so important to keep exploring and to keep finding new fungi?
Well, again, as I'm learning, fungi are amazing natural resources that have been underutilized. And so just like with the study of bacteria, which people know a lot more about, with fungi, they also can produce all these amazing compounds. There's undiscovered potential for them in a lot of different realms. So fungi have a long history of being exploited for their ability to treat not only other fungi, to kill other fungi, but to kill bacteria. They're also involved in statins for cholesterol. Psilocybin is being studied for potential antidepressant activity. There's lots of different uses for fungi. And for the most part, we really, we value and we gain a lot of amazing products from fungi. And we're actually now starting to look at ways in which fungi, since they're natural decomposers in our environment, that they may be able to degrade plastics. So one of my newest students is actually working on a plastic pollution project where we're screening fungi for the ability to break down compounds that are much more difficult to break down by the enzymes that we currently have. And so this is one of the resources that is sort of an unexpected find in our science, that we can actually utilize fungi not only to be just afraid of them, but what can we actually harness the power of fungi to do in our laboratory.
How many, like if you had to estimate what percentage of fungi in the world we've already learned about, what percentage do you think that is of all of the ones that exist?
I think I've seen figures like this, and they put it like we've probably surveyed about 3% of what's actually out there. Yeah, fungi are historically understudied, and there's so many different varieties. You could probably go into your background in your backyard and actually find a new fungal species.
Gusa's team has started to close this gap, and it began in an unusual way. In September of 2024, Hurricane Helene swept across the southeastern part of the U.S. and was particularly devastating to southern Appalachia, killing at least 250 people and leading to flooding that caused immense damage. In the aftermath, Gusa and her team visited the areas that had been flooded with one specific goal: to collect fungi.
And so these were isolated after Hurricane Helene. We went and did environmental sampling, and we've been able to curate this amazing resource that can now be used for potentially natural product discovery, and also to understand what kinds of mycotoxins or harmful allergens they might be producing that might be making people sick.
Gusa's lab collected hundreds of fungal samples from Black Mountain, North Carolina, an area that was hit especially hard by Hurricane Helene. The team has cataloged all sorts of important information, like the species, origins, and genetic sequence, to create a fungal biobank.
We have quite a variety. We wanted to show you just different phenotypes on the different plates.
We talked with Sam Schultz and Gabrielle Walker to learn more.
But basically what happened is we got a swab of all these different environmental samples, and my job is to go through and try to isolate out different species. And then once they're isolated out, this is a good example of them being isolated on different types of plates. I then go through and I have to identify them.
So you're basically starting this like big library.
It's a library.
Fungi.
Yes.
Okay.
Yeah. And we have a lot of species right now. I think we have about 65 in our biobank at the moment. All of them have been isolated and identified, 41 unique species, and out of them, about 10 of them are potential fungal pathogens.
What kinds of things are you looking for from the biobank as you assemble all these samples?
So we're trying to just basically find out the fungal abundance. The neat thing is that we can track which sample we have here, where the swab came from. And then the one neat thing too is with our fungal biobank, we also are recording the pictures as well. So this will be also very useful for people in the future to reference, making sure that when they do use our samples, they can look at our pictures and make sure that what they're growing looks like what we have growing here.
In addition to investigating which fungi might lead to infections, Gusa's lab will use the biobank to conduct research on potentially dangerous chemicals that fungi can make.
We have other researchers at Duke that are my collaborators that are going to be looking at what kind of toxins do fungi produce as they're consuming different materials. So when they're growing on things like drywall or on the surfaces of plastics, what are they actually producing as they metabolize? And so some of the things that they secrete into the air, in addition to spores, are things known as mycotoxins and also volatile organic compounds.
You know that musty, earthy smell in a damp basement? That's partly made up of VOCs cooked up by fungus. The effect of VOCs on our health is less clear, which is why Gusa wants to study them.
So this is sort of a whole new area of research that has been understudied again, and we really don't have an idea about what kind of health impacts result. But we certainly know that we already know of certain allergens that are caused by some of these fungi that are growing from indoor mold sources, but we want to learn more about mycotoxins and VOCs.
Having this biobank might even help us identify some of those fungus-fighting fungi we were talking about before. Or We may even find fungi that can help us in other ways.
So this is one of the fungal species that I'm working with right now to identify to be able to degrade plastic. And so from what I found from some of our preliminary results is that this species can degrade a polyester polyurethane polymer called Impranel, and it's doing really amazing. So what some of my next steps is identifying what enzymes are being secreted by Aspergillus that can degrade that particular polymer.
And we think that this is a possibility that we can go from natural disaster to natural discovery. And that's really exciting. I have to admit, like when our lab first started, it was kind of all about the doom and gloom. Oh, the threat of fungi and disease and, which is very serious and we really do care about it. But it was nice to have another side where we can also talk about the solutions that fungi might be able to provide. And so that's been really exciting and a nice balance in the lab.
All this is especially cool because the Gusa lab is still pretty new, and they're only just getting started. Gusa had a different path to becoming a research scientist than most. After earning her PhD, she spent a fair bit of time as a high school science teacher.
I got to be able to learn how to communicate science and really inspire, hopefully, the next future generation of scientists. And so I had this really incredible opportunity to come back a little bit later in life to be a researcher at Duke University, which has been always one of my dreams. And so now, instead of teaching in the classroom, I have a classroom in the laboratory, and I'm able to really mentor my students and really see them grow and thrive in their own right. So it's been a really exciting transition for me.
And as it turns out, when we got here, we realized she's already a SciShow fan. Did you ever use SciShow videos when you were a high school teacher?
I sure did. Yeah, Hank Green, you were a constant in my classroom. Yeah, so just the way that SciShow broke things down, it was able to really just augment the lectures I had in a fun and very relatable way with our students. So I know when I told my lab that SciShow was coming to our lab, they were super excited. They're like, "What? Are you serious? I can't wait to be a part of this." So yeah, we're really excited that you guys were able to come and visit with us.
We're so excited you guys joined our ecosystem, and now you and your research get to go be in other classrooms. It's kind of full circle, huh?
Yeah, absolutely.
The work that Gusa and her lab are doing is vital for helping us understand why and how fungi can be so dangerous to us, and even let us develop new medicines too, all of which could be very useful, whether we're facing down just a bit of mold overgrowth or a full-on fungal zombie apocalypse.
SciShow field trips are made with our friends at HHMI Tangled Bank Studios. We've come together to bring you face-to-face with researchers at the cutting edge of scientific discovery. You can watch more of Tangled Bank's science content at tangledbankstudios.org.